Sorry, we have some technical difficulties, but we're back on track. So in today's presentation, we'll introduce the concept of viscosity fingerprint. We'll give a brief review of VROC technology, and we will introduce and present VROC initium, which is the ultimate automatic viscometer. We'll present some of the first results that we have obtained with this instrument, and we will introduce a number of applications that you can tackle with this instrument. Viscosity fingerprint is a pretty broad concept. It basically includes the characterization of the viscosity of sample as a function of any parameter that you could possibly imagine. That could be shear rate, could be temperature, but it could also be so much more. And VROC initium has been conceived with the purpose of allowing you to characterize your sample in a high resolution thanks to the automation. VROC technology, which stands for Viscometer Rheometer on a Chip, is a hybrid of microfluidics and MEMS technologies. By combining a microfluidic channel with a MEMS pressure sensor array, we're able to measure viscosity. Thanks to its design, VROC technology features measurements of absolute viscosity, in contrast to index or apparent viscosity. It allows for the smallest sample volume requirements in the market, with measurements of viscosity with as little as 10 microliters. It provides with a very wide dynamic range in terms of both viscosity range and shear rate range that we'll discuss later. And thanks to its flow-through design, it renders high throughput. Additionally, our technology is fast and has superior repeatability and accuracy. Let's review how VROC technology actually works. We have this microfluidic channel, and we can pump a fluid through it at a given flow rate. Since we know the dimensions of the channel, we can determine the shear rate, which is going to be proportional to that flow rate. Now, the pressure sensor array in the bottom of the channel, it's going to allow us to characterize the pressure as a function of position. If we have that, we can characterize the pressure drop through the channel, and that's going to be proportional to the shear stress. Well, by definition, viscosity is the ratio of those two parameters, shear stress and shear rate, and that's how we measure it. Now, I hope you guys are ready, because it's here, our new product release, VROC initium. You can see it as a whole for the first time. Here is VROC initium made unit, and then VROC initium with the auto sampler. We have prepared a video explaining how the system and showing a little bit of how the system works. Unfortunately, GoToWebinar doesn't work very well with high quality videos, so it might get stuck a little bit as we show it to you. But the video will be available on our website, and you will be able to play it at a higher quality. So let's see how this beauty works. VROC initium main unit, you can see it now on the screen. It features custom 40 vial rack and or alternatively 96 well plate. The auto sampler allows you to load the samples in a preselected order. So any formulation that you have prepared is going to be in this vial rack. and you're going to be able to load it into the auto sampler syringe. You can load as little as 10 microliters of sample for a measurement of viscosity, and it will be automatically placed into the main unit itself, as you can see right now. So it has been designed for the auto sampler to be able to load a bubble-free sample into the main unit. Once the sample has been loaded, the auto sampler will go back to the cleaning pour position where the auto sampler syringe will be prepared for the next measurement. And you will be set to start your measurements of viscosity. You have your sample load, and you can prime and then do your measurements of viscosity as a function of shear rate and as a function of temperature. Once you're done with your measurements, the system will clean automatically all the flow paths to allow you to test your next sample. You can see on the left the cleaning solvent bottles. This automatic system will allow you to characterize the viscosity fingerprints of your sample fully. Right. Let's summarize some of the key features. First one is it's a high throughput automatic viscometer rheometer. So all functions are automated. Sample loading, testing, and cleaning and drying can all be determined in a testing recipe and doesn't need attention from the user. It can measure viscosity with as little as 10 microliters of sample. It's compatible with a 40 vial rack and a 96 well plate, but it's also it allows for customization in this sense for other types of racks. It allows you to measure viscosity as a function of shear rate and temperature on rate and temperature sweeps that can be configured into the system. And it has two built-in temperature control that are independent. One is for the system itself, so you will control the temperature of the sample inside the syringe that is in the main unit and the flow cell itself from 4 to 70 Celsius degrees. And the other one is on the sample rack that will allow you to control temperature between 4 degrees and slightly higher than body temperature. Here's another view of the instrument. You can see the different components: auto sampler, main unit, a status LED indicator. On the left side, you can see the three different solvents that are used for cleaning. You have a primary solvent that will be compatible with each specific type of samples to allow for cleaning. An optional secondary solvent that can be used to speed up the process in some cases, and a dry enhancer that will enhance drying or speed up the drying steps. Here's a top view of the instrument with a somewhat higher level of detail. You can see the 40 vial rack or the 96 well plate where your samples will be placed. The auto sampler will come and load the samples from here and move them into the main unit pumping system. It will load them into this syringe through the 100 microliters injection port, and then the auto sampler will move back to the cleaning port where will be prepared for the next sample. For the very high shear rates, you have an additional injection port for a 5 ml syringe where you can load 5 ml of sample that will allow you to test those very high shear rates up to 1.6 million. Once you have your sample loaded, you can run it different shear rates and different temperatures through the cell. You will follow this flow path, and you will get your measurements of viscosity or the viscosity fingerprint of your samples. Once you are testing, you have two alternatives. One is to direct your sample straight to waste, and one is to direct it to a recovery reservoir that we will explain in more detail later. Cleaning is achieved through the different solvents. So, they will be automatically, it's automatically featured by the system. You will pump the primary solvent through the different flow paths that you need to clean, and this all be automatic. If you have a secondary solvent, that will follow the primary one, and then the drying enhancer to speed up drying of the system. Powered by VROC technology, VROC initium enables a very wide dynamic range of operation in terms of both shear rate and viscosity. You can measure shear rates from 1 inverse seconds to 1.6 million inverse seconds, and viscosity ranging from 0.2 centipoise to 100,000 centipoise. That you can achieve by combining 4 different types of chips, A, B, C, and E, and different channel depths ranging from 20 to 300 microliters. That allows our system to tackle a large number of applications, going from flow coating and mixing to high shear applications of engine oil, and paying special attention to biopharmaceutical applications such as injectability and stability of protein therapeutics. So let me summarize the dynamic range of the instrument. It covers viscosity range from 0.2 to 100,000 centipoise, 1,000 centipoise with the auto sampler, and the shear rate range is from 1 to 1.6 million inverse seconds. This allows you to go about your sample testing in several ways. One of them is you can rank your samples at a fixed shear rate in terms of viscosity. You could also measure viscosity as a function of shear rate and obtain the viscosity fingerprint of your sample or the viscosity curves. You could also measure high viscosity at high shear rate, which is important for applications such as injectability. Another alternative that you have is to just run automatic mode. That will automatically select the appropriate shear rate and volume required for your sample and enable a quicker and easier analysis for your results. Right, now it's time for us to show some of the first measurements performed with the instrument. VROC initium features astonishing repeatability and accuracy that are required for applications such as measuring intrinsic viscosity, where the viscosity dependent of very dilute solutions is going to require these very precise, very repeatable measurements. Repeatability is 0.5% of reading, and accuracy of the instrument is 2% of reading. On the figure on the top left, you can see how you can repeatedly perform measurements of viscosity, obtaining the same results within 0.5%. On the two tables below, you can see results for viscosity measurements of standard 35 oils for 2 different types of chips, and you can see how impressive the accuracy results are. As we mentioned before, you can perform measurements with VROC initium with as little as 10 microliters. This is very impressive. You can see in this figure how with 10 microliters you can get a highly accurate measurement of viscosity. VROC initium will come initially with 2 configurations: standard configuration that will allow 10 microliter measurements of viscosity, and a high shear configuration for applications such as inkjet printing that will allow for those highest shear rates, will require some additional volume for those. In future releases, we'll have an extremely low volume configuration that will further reduce the sample volume requirement below that 10 microliters. Here we present some of the first results for protein solutions. On the plot on the left, you can see shear rate sweeps for BSA in PBS solutions at different concentrations. On the right, you can see how for the same samples, BSA in PBS, it's in green, you can see the viscosity dependence on concentration. Now, you might want to study what's the effect of your buffer solution in the solution. And you can see how, in this case, if we test BSA in water, we have a different, somewhat stronger dependence of the viscosity on concentration of the protein. You might also want to study how the protein size is going to affect the viscosity as a function of concentration. We have tested some immunoglobulin, and you can see how the dependence of viscosity and concentration is much higher. All this allows you to characterize the viscosity fingerprint of your samples with a very high resolution that is enabled because you can take large amounts of data thanks to the automatic system. Finally, we wanted to show some results for the characterization of non-Newtonian fluids. On the figure on the left, you have Shandong gum solution 0.5%, and as you can see, this system is strongly shear thinning. What that means is that the viscosity strongly depends in shear rate, and in fact, it decreases as you increase the shear rate. To be able to characterize these kind of samples requires a very good control over the shear rate that you are subjecting them to. This is key for applications such as injectability, where you need to measure the viscosity of your samples at the characteristic shear rate for the injection process. I would also like to point out that we have 6 measurements here that are the result of average of 3 repeats, so that's 18 measurements. We perform these measurements with a single syringe, and we still have quite a bit of sample left. So you can test more than 20 shear rates with a single 100 microliter syringe. Well, if that's not enough for you, let me present the next feature of VROC initium. You can retrieve your sample. So you can load your 100 microliters or the volume that you have available of sample into your system, and you can test it. You can measure at some specific shear rate, get some repeats at some specific temperature, and you will direct it, instead of to the waste, you will direct it to the recovery reservoir. After that, after your testing, you can recover your sample back into your syringe. And here you have 2 options. You can fully recover your sample, so you can extract it and go ahead and do some other testing that you might be interested in, some different property. Or you can further test viscosity on the same sample volume. So you could test back and forward for many measurements of viscosity at different shear rates and different temperatures with one single volume loading. That will allow you to study shear stability of your sample, as you can test the same sample back and forward many times. It will allow you to test thermal stability. You can test your sample at a given, let's say, shear rate sweep at a given temperature, and then you can modify that temperature and test it again, and so on until you find some, I guess, critical temperature where the properties of your sample change. It also, as I mentioned, will allow you to recover your sample to do some testing of some other different property. Now we want to give you some good estimates of the characteristic measurement times for the instrument, VROC initium. For those of you who are familiar with an instrument, a single measurement of viscosity time is usually less than a minute. So that's really quick. Cleaning and drying procedures for VROC initium can take from 6 to 10 minutes. So using those, you can see how testing a full 40 vial rack of samples will take you just slightly shorter of 8 hours. And testing a full 96 wall plate will take between 12 and 16 hours. So what you need to remember though is that all that testing is fully unattended. So you will prepare your samples, determine your testing recipe with loading, testing, and cleaning protocols, and then you can press run, walk out, and come back the next morning to analyze your data results. VROC initium features an XYZ autosample, which allows you for these high throughput automatic viscosity measurements. It is compatible, as I mentioned, with 40 vial racks and 96 well plates, but it also has a teaching mechanism in the software that will allow you to use a different variety of plates or racks for your sample. The autosample features a 100 microliter syringe with an 18 gauge needle, which allows you to measure these high viscosity liquids in an automatic fashion. Right, let's go through the workflow for the high throughput testing. First, you will need to prepare your samples, and you will place them in either a 40 vial rack, 96 wall plate, or a customized rack that you may want to use. Next, you need to determine what's your recipe, and that includes sample loading protocol, measurement protocol. You want to do a single point measurement of viscosity for each of your samples. You want to do a shear rate sweep or a temperature sweep or a combination of all of this. It's really up to you. And then you will determine the cleaning protocols for each of the samples in the 96 wall plate. Once you have your recipe, you are ready to perform your measurements. So you will hit the run button, go home, let the system do the work for you. And when you come back the next day, you will be able to use all the software capabilities that we're going to provide for the data analysis, report, and export of data that you have collected in an unattended way. All right, so let's go back to the concept of viscosity fingerprints, because we want to show you what the real capabilities of this instrument are and what's going to allow you to do. So let's say you have a protein and you want to test a number of formulations as a function of concentration. That can be very interesting by itself, and you can do it with VROC initium with high resolution because you have an automatic system to test your samples. Well, there is a number of parameters that you might want to look into too. You can look into the pH effect on your formulation, and you can test that. You can see, as we showed before, how different buffers have present a different environment for your proteins, and that can have an effect on viscosity of your formulations. Well, you can test that too. Different excipients. You can might want to add sucrose into your solutions to make the proteins more stable. You can test that. You can test different concentrations of different excipients and see the effect on your formulations. You can test different ways of the nature your proteins. You can test the effect that temperature has in the viscosity of your solutions, how viscosity will increase as your protein denatures, and you can test the effect of different denaturants and their concentration on the viscosity of your samples. And you can pretty much test any different parameter or any different change in the environment in your formulations that you can possibly think of. That's going to allow you to test the viscosity fingerprint of your samples, and it's going to allow you to tackle numerous applications. One example is injectability. You can measure high shear of all these kind of different formulations and variations, and that will allow you to fully characterize your samples. Another alternative that you have is, since our instrument is extremely repeatable, you will be able to measure intrinsic viscosity, and that will allow you to characterize the molecular size of the proteins in your formulation. That will give you additional information about all the processes taking place as you change all these parameters. And you can also study the stability of your different formulations as you change pH, temperature, or anything you can imagine. So let me summarize the measurement capabilities and applications for VROC initium. Is the first high throughput automatic viscometers. It's a high throughput screening system for viscosity that allows you to study things such as injectability for protein therapeutics, stability for protein therapeutics, kinetics of chemical reactions, solubility, intrinsic viscosity for molecular side, but the list is limitless. High shear, anything really. Now there is some additional features. One is that the system is 21 CFR Part 11 compliant. There is a number of tools that will be included in the software, which has a database with user access control. And those tools are designed to allow you to have easier and quicker way to analyze your data. You have data analysis tools such as overlay, comparison, curve fitting, pass and non-pass criteria, model fitting, and so on, that will allow you to prepare reports on your results, but also understand what's the viscosity and the rheological behavior of your samples. With that, I would like to thank everyone for joining this webinar. I hope you guys enjoy the first sneak peek to our newest product. All the materials that we have shared today will be available in our website shortly and will be provided to you. And with that, we will take some questions from the audience. And in the next, you have our contact information if you can think of any questions after the webinar that you want to transmit to us. Thank you.